let's look at the first, well, one of the most important maternal genes, which was bicoid. And the reason that bicoid has actually enjoyed a lot of work in the last 20 years is that it appears to be a morphogen, and it was the first morphogen that was actually identified, and it was discovered in a maternal effects screen from Janne Nusslein-Volhard's lab. And when she looked at and her students looked at the phenotype of a larvae in a bicoid mutant, what they found was severe anterior and thoracic patterning defects. So many of the anterior and thoracic segments were completely screwed up, just looking at the larval cuticle. And when one of her students, Wolfgang Drever, had cloned and the bicoid gene and generated antibodies against the bicoid protein, he found that after fertilization, the protein was set up as a gradient in embryos. And this was a gradient that could be quantified. It looked like an exponential gradient. And so that fit perfectly with the notion of morphogens providing some type of information that nuclei could read out at different concentrations and acquire different types of cell fates. Now, this slide is just to illustrate how extensive the patterning defects were in bicoid. The top part of the slide shows the wild-type embryonic segments with head segments and mandibular segments and thoracic segments and abdominal segments. And all but a number of abdominal segments have their patterning completely screwed up in a loss-of-function bicoid mutation. So the inference is, of course, that bicoid function is required for the patterning of more than the front half of the embryo, and therefore plays apparently a major role in setting up the anterior-posterior patterning in Drosophila. Now, one of the reasons that bicoid has such a severe phenotypic effect is that it's known that it turns on a number of different target genes. So when bicoid isn't working, these target genes are not expressed appropriately. And these target genes have different patterns of expression along the AP axis, again consistent with the notion that they may be reading out different concentrations of bicoid. And we're going to look actually pretty carefully later on to see if that's actually correct. Now, when Janne's lab looked at the localization of bicoid messenger RNA, which was at the anterior, and the gradient that was formed after fertilization of the bicoid protein, she and Wolfgang Drever came up with a relatively straightforward model called the synthesis, diffusion, and decay model that would explain how that gradient could remain stable so that nuclei could read out a constant amount of bicoid over developmental time and acquire the appropriate cell fate. And next week we'll be looking very carefully at how well this model is held up.